I'm Paul Hanson from Oak Ridge National Lab, and I'm pleased to talk to you today. As you can see, I've modified my title from the schedule and consider it ruminations rather than a description of knowledge gaps on ecosystem functional responses to anticipated climate change. Today I'm going to take a few slides to take a quick look back at other experimental work that we've done in the past and thoughts that got us there. I'm going to provide thoughts on quantitative descriptions of key climate or environmental change drivers, which I think are important in defining knowledge gaps. I have a number of slides on process level knowledge gaps that we continue to need to strive to understand, and a slide on important overlooked processes. Back in 2007, discussions at Oak Ridge National Lab suggested that we should be seeking leadership in experiments and observations for developing process understanding necessary for forecasting long-term climate change responses of ecosystems. The researchers were expected to provide society with confidence in determining safe stabilization levels for greenhouse gases in the atmosphere and the impacts from climate change. Planned experimental manipulations would focus on ecosystems that were hypothesized to produce key greenhouse gas or albedo feedbacks to the climate system, and for those that provided important goods and services to society. We did emphasize rapid translation of new mechanistic discoveries into process model simulations, the parameterization of models using data assimilation, and the visualization of climate change. Back in 2008, DOE supported another workshop or a previous workshop on ecosystem experiments. The topics of that workshop covered climate change impacts on ecosystems or response studies, terrestrial ecosystem feedbacks to the climate system or forcing studies, discussions of model experiment interactions, discussions of the priority ecosystems and climate change treatments that might be needed in the future, and further discussion of new technologies and measurement needs. In that, of the group of us participating this week, 4 of us were present then: myself, Alan Knapp, Rich Norby, and Alistair Rogers. And since that time, the DOE and other agencies have supported any number of precipitation change or drought studies, warming studies like Spruce and Be Forewarned, the NG Arctic and the NG Tropics observational regional initiatives. Coastal research has come on recently, and DOE has pushed in recent years for closer ties via MODEX activities to the E3SM model. My point of putting this slide up is that to point out that workshop concepts and conclusions do and can and do have an impact. I've included this graph to show that over time, the research community's focus has changed. The graph is from a retrospective review that Anthony helped me write for Global Change Biology's 25th anniversary issue. And it points out that, we've gone from focuses that largely on elevated CO2 in the past and ozone in the past to big emphases these days on disturbance and warming. But I think it's important to point out that all of the driver environmental variables are still in play. And in my opinion, I think that all need to be considered in efforts to understand integrated ecosystem responses to climate change. And we should keep them in mind as we continue this week's discussion. I'm now going to transition to a few slides on what I'm calling understanding environmental changes in a numerically quantitative manner. And this is predicated on my thoughts that ANOVA analyses in experimental studies have produced good results in the past, but they're largely insufficient to me and unsatisfying as they don't provide a quantitative bridge to prognostic model results. Deciding that something happened in a statistical manner is very different than saying what happened and how big the results were. So I think we must move beyond determining if an effect can be declared significant to understanding how and when the effects occur along a numerically defined scale of change. I think emphasizing the quantitative nature of environmental manipulations is an important consideration, especially when choosing and designing new manipulations and subsequently for the interpretation of published results. What I mean by being numerically quantitative in the context of elevated CO2 is that we simply need to be cognizant of the CO2 concentrations that are involved. Correlations to biological response over historical changes are fair enough, but we need to recognize the timing and the concentration ranges involved. Manipulative studies for forced increases in atmospheric concentration, what has been done in the FACE study and other chambered studies, also can be used and compared, but we need to know whether one study is at 150 parts per million or another one at 250 or 400 or 500. I am concerned about published results of recent limited changes in CO2 and their correlations to some sort of response, because such conclusions seem inconsistent to me with somewhat modest impacts associated with larger scale experimental manipulation differentials. In the context of warming studies and my desire to have us be numerically quantitative, I'm making this point because recent reviews of experimental warming studies have attempted to lump together meta-analyses so that they can draw general conclusions about global change under a warmer environment. That's fair enough, but I think we would be better served to retain the details of the nature and magnitude of the warming manipulations so that we can interpret or extrapolate warming study responses. The bulletized list below here just suggests the range of and kind of warming studies that are out there, with details that should be conserved as we compare and contrast studies. With respect to temperature change or warming studies, it's clearly important to understand carbon cycle phenomena, gross primary production, and ecosystem respiration, but we shouldn't lose sight of other things that happen with warming, and that is changes, for example, in freezing depth, snow depth, cold hardiness, and even periodic frost damage under a given warming scenario. We should ask the question, how does the warming impact available soil water or water table depths as well? The graphic simply shows seasonal temperature pattern graphs for the spruce study at 3 different temperature treatments, showing that when you get warm enough, ice and snow disappear from the system, and that changes the nature of biological response. The third example I have is some thoughts on drought in a numerically quantitative manner. Drought and precipitation change are certainly often compared across studies without consideration of climate zones, ecosystem types, and of soils in an attempt to come up with general conclusions. I think we should do better. In the context of organismal response, that is plants or microbes, the unifying concept should be water availability. In models, this is often expressed as available water as measured by percent soil moisture within a defined soil depth or depths, and soil texture or rooting depth may or may not be considered adequately. I think water potential represents the best unifying metric for the characterization of short-term water availability to organisms, and we've found that it also serves as a very useful cumulative metric for seasonal drought severity in the interpretations of our past precipitation manipulations and vegetation responses at the Missouri flux site. Now I finally get to some thoughts on knowledge gaps. The first one's related to ecosystem forcing feedbacks. Certainly global ecosystems play an important role in moderating the climate system through interactions with the carbon cycle controls on greenhouse gases and through surface energy balance changes driven by land surface composition. Research has clearly made advances in our understanding of ecosystem functions in all of the above carbon, water, and nutrient cycles. And this is indeed where we have spent and need to spend much of our time and effort. But I ask the question, have we advanced our capacity to project future land cover and function? And I guess I do not have a lot of confidence in projections of what land use patterns will be in the future, nor do I think we are able to project successional changes under future environmental change. A couple of points on knowledge gaps with respect to ecosystem impacts. I think we spend most of our effort here when we study environmental changes, defining impacts on processes, organisms, and element cycles. And we've come a long way. Two things that remain elusive to me are understanding carbon allocation to growth and understanding and predicting mortality. With respect to carbon allocation, I'm really struck with the idea that we have hypotheses, they are being tested, but have we ended up with a universal algorithm that works across ecosystems? And similarly for mortality, do we have mortality driver algorithms that work, and do they vary by ecosystem? I think they probably ought to. I believe there exists a knowledge gap for scaling the rates of soil carbon decomposition from warming across peatlands that exist from permafrost to temperate latitudes. Carbon stored in permafrost conditions is frozen. As such, it retains a labile characteristic of newly formed plant material. When that material is thawed and perhaps exposed to oxygen, the organic material is consumed and re-emitted by microbes at a rate characteristic for the permafrost system. Organic material stored in temperate peat has undergone thousands of years of gradual change associated with the loss of the most labile components. When it is warmed or exposed to oxygen, its rate of decomposition may therefore be very different from that of the permafrost system. I think we need a fundamental plan to scale such results across extant peatlands. Here's a graph to break up all of the words in my chit-chat, but this shows the results from the spruce study, the linear declining loss of carbon associated with the warming treatments from 0 to plus 9 degrees C. And I've overplotted that some results from the Ted Scher group's carbon losses with snow depth induced warming. And it turns out that maybe the scaling wouldn't be that hard because the data actually tend to overlap at this point in time. But we need to study this further, and we do need a plan for extrapolating such results to other peatlands. An important knowledge gap that we almost completely ignore is the role and projection of future impacts of pests, pathogens, and invasive species. I think you could make an argument that the greatest impacts to forest composition and perhaps function as well are the influence of pathogens and invasive pests. We certainly have little capacity to model their occurrence or their impact. Pests, there's a whole list of the ones that are available that we should be concerned about. We don't know how they're going to respond under climate and atmospheric changes, but we should be considering that. Similarly for pathogens, or even the interactions between the presence of pests and pathogens and our environmental drivers with global change. While these topics may not be considered priority research for the Department of Energy funding, it's certainly often overlooked and may have major impacts on carbon, water, and nutrient cycles from time to time. And finally, my last slide is a comment on model data and experiment integration, something we absolutely must have, but I also suggest that we shouldn't spend significant time or effort to develop a new manipulation and the associated measurements unless we have a solid plan to use and interpret that information. To me, that means models will be used to plan, interpret, and potentially improve experimental efforts. Expending effort to study curiosity research questions might in fact undermine progress on our understanding of important carbon, water, and nutrient cycles that we are trying to predict first and foremost. However, if we're going to build something new, we should not presume that we have already identified all important processes important to environmental change interactions, and allowing colleagues to address their curiosity questions within existing or new research facilities is always worthwhile.